Hello students, welcome to Biosmart Academy. In today's lecture, we will continue our lecture series on isoelectric point. Okay, so in the first lecture of isoelectric point, we discuss about the basics of isoelectric point. Okay, so please go and watch the first lecture of isoelectric point because this first lecture will help you to understand the upcoming lectures. Okay, as well as today's lecture. So what we will going to discuss on today's lecture, that is how to calculate this isoelectric point for non-polar amino acid. Okay, for non-polar amino acid. In today's lecture, we will see only for non-polar amino acid. Okay, then in the upcoming lectures, we will see polar amino acid as well as for some special amino acid. Okay, so let's start today's lecture, that is isoelectric point for non-polar amino acid. Okay, for non-polar amino acid, how can we calculate the isoelectric point for non-polar amino acid? Okay, so first of all, I just want to say you that please take a copy and pen and please draw with me, please calculate with me, then you will get a clear idea. Okay, so first of all, take an example of non-polar amino acid, that is alanine. ALA, its single letter word is A. Okay, you have to remember single letter word for each and every amino acid, and I made a trick videos on amino acid lecture series. There will you find 4 lecture series on amino acid. Okay, and that lectures were very beneficial for your preparation. Okay, please go and watch that. Okay, so we take the example of alanine. Okay, so what will be the structure of alanine? This will be the structure of alanine. So this is the structure of alanine and this is a side chain CH3. Clear? So this is a side chain of alanine, that is R. Okay, now this structure of alanine is at pH 1, remember it is at pH 1. Clear? Now to understand the isoelectric point, you should know three things actually. You should know 3 things. First, pK1 value for acidic part, that is COOH part, that is we take as an average 2. Please remember this, we take an as average pK1 value is 2 for COOH, and we take an as an average pK2 value for NH3 is 9. Okay, so first remember this, we take pK1 value is 2 for COOH and we take pK2 value 9 for NH3. Okay, so this is the first important thing you should remember. And another important thing you should remember is the when we have to take protonated and when we have to take deprotonated. Okay, so what is protonation? Protonation means addition of hydrogen. Suppose this is CO minus, and if we add hydrogen into it, that is H plus, if added, that is protonated, it becomes COOH. So this is protonated, addition of H plus. Okay, and what is deprotonated? Just look at this. Suppose this is COOH, and if we remove the hydrogen, then what happens? It becomes CO minus. So this is basically deprotonated. Clear? Okay, now so this is pK1 value. Okay, now if we go below this pK1 value, that is from 0, 1 and 2, then we will take as COOH. That is we take the protonated form. If we go below this pK1 value, that is 0, 1 and 2. Okay, now if we go above this pK1 value, suppose 3, 4, 5 up to 14. Okay, so if we go above this pK1 value, that is 3, 4 up to 14, 3, 4, 5, 6, that is up to 14, then what happened? Then we have to take this COOH as CO minus. Clear? Okay, so this is one concept. And same concept is applied for this part also. Okay, if we go below this, that is 0 to 9, okay, that is 0 to 9, then it will remain as NH3 plus. Okay, and if we go above 9, that is 10 to 14, then this NH3 plus become NH2. Clear? So this is one concept regarding the calculation of isoelectric point for non-polar amino acid. Okay, now I will calculate the isoelectric point for alanine. Just look at this. Okay, so this is a structure of alanine. This is a structure of alanine. Okay, and I already told you that this is exists at pH 1. Okay, we have to go from pH 1 to pK 1, pH 1 to pK 1 to pK 2 to pH 14. Clear? So first we have to calculate pH 1, then we have to see pK 1, then we have to see pK 2, and then we have to see pH 14. Okay, here no pKr is present. pKr we see in our next lecture. It is in case of ionizable groups, or you can say polar groups. Okay, so and remember one thing, we just take this value according to pH. pH 1, that is lowest pH, pK 1 pH is what? 2, because this average value we take as 2, as I already mentioned. Okay, then we take the pK 2, its pH is 9, that is value is 9, and it's 9, that's why we take pH pK 2, then it's 14. So we arrange this in a pH manner. Okay, from 1 to 9 and 14. Okay, now so this is the first step. So in pH 1 you will find the same structure. Okay, and in pK 1 also you find the same structure, that is this structure. Okay, that is in COOH form and in NH3 plus form. Okay, clear? Why in pK 1 you find the same structure? Because its pK 1 value is 2 and its pK 2 value is 9. So in pK 1 you find the same structure. Okay, clear? So at pK 1 the charge is what? The charge is plus 1. The charge is plus 1. How we can tell the charge is plus 1? You can see that in pK 1 this is neutral and it contains plus charge. So this is plus 1. So in pK 1 it contains plus 1 charge. Clear? Now when we move from pK 1 to pK 2, okay, when we move from this pK 1 to pK 2, then what happened? The pK 2 value is 9. So what happened? So this COOH becomes COO minus, because we move above 2, that is 9. Okay, so this structure become from pK 1 to pK 2, this structure become like this: C alpha, COO minus, NH3 plus, this is the R group and this is the hydrogen. Clear? So now you understood why this becomes CO minus, because we take as pK 2 9, and we know that above this 2 it become deprotonated, that is become CO minus. That's why it becomes CO minus, but it remains as NH3 plus because its value is 9. That's why it remains as NH3 plus. Okay, so here the charge become what? It contain minus 1 charge and it contain plus 1 charge. So plus minus, that is charge become 0. Plus 1 minus 1, that is charge become 0. So at pK 2, from pK 1 to pK 2, the charge become 0. Charge becomes 0. Clear? Now when we see the pH 14, when we see at pH 14, what happened? So in this case also, this NH3 become NH2. NH2, why? Because it is above 9. pH 14, that is 14 above 9. So I already told you that when it move above 9, then it become what? Then it become NH2. NH2. Clear? So the structure at pH 14 become what? this become like this. Okay, this remains CO minus because above 2, that is pH 14, and this become NH2. Okay, because it is above 9, that is pH 14, and this is a hydrogen and this is the group. So in this case, what happened? In this case, the charge is only -1. So the charge is only -1. Okay, that at pH 14 the charge is only -1. Why -1? Because this CO minus group contain only charge, it become neutral. Okay, clear? So this is a concept. Now I hope that you understand this concept. Okay, now, so what is isoelectric point? We all know in our basic lecture I already explained the point where the charge becomes 0. The point where the charge becomes 0 is called isoelectric point. That is, the point between pK1 to pK2 is the point of isoelectric point. Clear? So for non-polar amino acid, the calculation for isoelectric point is Pi just equals to pK1 plus pK2 divided by 2. Then you will get your answer. Okay, for isoelectric point. Clear? So this is a formula for non-polar amino acid calculation for isoelectric point. Okay, and you get to know from this lecture that how you get this formula. I already explained this. Okay, now if you don't understand this, then please repeat the video, please note on your copy, then you will get clear idea. And if you still not understood, then please comment me, I will try to clear on the comment box. Okay, so that's all for today's lecture, that is calculation for isoelectric point for non-polar amino acid. I hope that you find this lecture helpful. For more lectures, please share BioSmart Academy and please subscribe our channel. Okay, it will motivate us to make more and more videos. Okay, so on our next lecture, we discuss about the calculation of isoelectric point for polar amino acid. Okay, so till then, thank you. Stay safe, stay happy and stay healthy.